So today we're going to be taking a look at case studies 2 and 9. And we're going to start with case study number 2, which has to do with CD40 ligand deficiency. So question number 1, CD40 is located on blank cells and CD40L is located on what other types of cells? So CD40 is found on B cells, macrophages, dendritic cells, all kinds of cells, mast cells, some epithelial and endothelial cells, whereas CD40L is located on T cells specifically. Now, what makes a B cell produce IgE antibodies versus IgM antibodies? So, cytokines IL-4 or IL-13 are required for this, but there also has to be CD40, CD40L interactions between the B cell and a T helper cell. Question 3, once IgE is produced from a clone of B cells, can you make IgG? Why or why not? What about IgA? So once class switching has taken place to IgE, the gene for IgG will have been removed from the genome in these cells. So there is no class switching or isotype switching possible from IgE to IgG. IgG is upstream from IgE. However, IgA, the gene for IgA, is downstream from IgE, which means that class switching or isotype switching from IgE to IgA is possible. So how did doctors determine that Dennis did not have CD40L? And is there another way to figure this out? So what they did was they activated the T cells so that what that would do is it would trigger the presentation of CD40L on their cell surface. And then they took soluble CD40 that was fluorescently tagged, added that in to these cells to see if these T cells that are activated and should be presenting CD40L, see if they'll actually bind the soluble fluorescently tagged CD40. And then they used flow cytometry to check, and the T cells did not bind CD40. And another thing that they could have done to figure this out would be to sequence the genome. Moving on to number 5, why didn't Dennis have an issue isotype switching from IgM to IgD? IgM and IgD is not, there is no isotype switching taking place there. IgM and IgD are results of alternative splicing. So since there is no isotype switching taking place, there's no need for CD40 signaling. Number 6, why did Dennis make antibodies against blood group A and B antigens, so our ABO antigens for blood typing, but not against tetanus toxoid, typhoid O, or typhoid H? Would you expect him to make antibodies against the protein capsule of Streptococcus pyogenes? So the A and B blood group antigens are sugars, so those are T cell independent antigens. The B cell can respond to those antigens without the involvement of a T helper cell. The other antigens mentioned, such as the tetanus toxoid, typhoid O, the Streptococcus pyogenes, those are all protein antigens. So those are T-cell dependent antigens. It's going to require that CD40, CD40L signaling. Why are IgG antibodies more important for protection against pyogenic bacteria than IgM antibodies? So these are encapsulated bacteria. They're resistant to destruction by complement. They're also resistant to phagocytosis. In order for them to be phagocytosed by neutrophils and macrophages, they need to be opsonized. And IgG are opsonizing antibodies. IgM are not. And so IgG are more important for protection against these types of bacteria than IgM. IgM can neutralize, but they don't opsonize, so they're not going to increase phagocytosis. All right, moving on to case study number 9, which is about DeGeorge syndrome. So what is the most common cause of this syndrome? What is significant about the location of the genetic abnormality? So the most common cause is a 3 megabase deletion on chromosome 22, a specific region. And primarily what is notable about this region is it contains the gene for TBX1. The TBX1 protein is a transcription factor that's involved in tissue and organ development. So without a functional TBX1, this is where we see abnormalities in the heart, the thymus, the parathyroid gland in these patients. And there's also more than 35 genes in this area, but TBX1 is the gene of note in this syndrome. What is FISH? Number 2. And how can it be used to diagnose DeGeorge syndrome? When will FISH not tell you if a patient has this syndrome? What is another way to make this diagnosis? So FISH is fluorescent in situ hybridization. It shows if there's a lack of a region of DNA. There are these fluorescently tagged DNA or RNA probes that will detect regions on chromosomes. So they would have a probe that's specifically for this region that's suspected to be deleted, and then use it to see if that region of chromosome 22 is actually present or not. What it can't tell you is if there's like a point mutation in TBX1 or in some other gene that affects thymus development. And another way to diagnose that would indicate the presence of other mutations would be to sequence the genome. What does hemizygous mean? It's only one functional copy of a gene is present. Excuse me. And, for instance, in biological males, any genes on the X chromosome are hemizygous for those individuals. And question 4, bone marrow transplantation is the standard treatment for many immune diseases, but not DeGeorge syndrome. Why? So with these other, especially, we've seen this in, I think, a couple of the previous case studies as well, where there's bone marrow transplantation to replace the, immune cell progenitors in the bone marrow. But there's no deficiency in these patients in the formation of immune cells. Progenitors can be produced just fine. The issue at hand is that there's no thymus, and the thymus is needed for proper development of T cells. So basically, these patients, especially those with the complete syndrome, need a thymus transplant. And thymic transplantation from unmatched HLA donors is usually done in DeGeorge patients. Why does this work? So the reason for that is that the issue of unmatched HLA in transplantation is due to detection of that unmatched HLA by T cells. There aren't functional T cells, so there's no recognition of unmatched HLA going on in these patients. And number 6, DeGeorge patients have a higher risk for autoimmune disorders. Why is this the case? So there's no negative selection going on for autoreactive T cells, and so those T cells are not deleted. Therefore, even though these patients have very low levels of T cells, the T cells that are there are potentially, some of them can be potentially autoreactive, and if there's any clonal expansion of those cells, that can result in an autoimmune disorder. And that's it. See you for the next one.